Okay. Hello, everyone. Welcome to the third session of the NPTEL course Data Analysis for Biologists. I hope you can all hear me. Just confirm. All right. Thank you. So I'll start by sharing my screen. Before we begin today's sessions, do you have any question? Any doubt from any of the sessions before or anything about the course? Nothing? Your voice is not clear. Can you maybe write in the chat box? Okay, so should I start? So we have had 2 live sessions until now where we have discussed the first 3 weeks of this course, and these recordings have been uploaded to YouTube. So today we'll be discussing the week 4, which is about how to visualize data using R and RStudio. And just to clarify that in the exam, the final exam, which is in March, there would not be any questions that would require the use of RStudio or of any type of matrix calculator which is available online. Instead, there would be questions like which would test your knowledge of R and how to write these codes, like you would have seen from the past 2 assignments. And as well as there'll be simple matrix calculations, which would just require you to know some formulas, like the formula for 2 by 2 determinant or rules and so on. But they won't require you to. Hello?
Yes, and
Yeah. Okay. And also an online scientific calculator will be provided to you. All right. So, okay. Now, so before we start, so in each of these codes, sir has used some CSV files and some R scripts. So I hope you have all been able to access those. They are available below each video on NPTEL, and they're also available on sir's GitHub repo. So do you have any, have you faced any problem accessing these codes and these files? If so, I can show you through. Otherwise, I also demonstrated this in the last video, so you can take a look there as well. No is for no doubt or have not been able to access. Could you just, no issues and accessing the file. All right, great. So now this session is specially designed so that if you have any doubts, any small doubt, big doubt, you can ask. All right, so please don't hesitate in asking anytime as we move on to this lecture, okay? Okay. So this is just the basic. I hope after last session, you all have R and RStudio installed. This is just a recap. I hope nobody has faced any issues with the installation. Okay. So again, like the last time, we will directly go to RStudio. So I have a script ready with me. So which I've named script underscore week4.r. It's an R script, so the extension is .r. I'm just going to click this, and by default, the computer recognizes that you need to open RStudio for this. Otherwise, I could have opened RStudio and went in the file, and then if I want to open a new file, or if I want to open like an existing file. Okay, so we have a lot of things planned for today's session since a lot was covered in this week. So I'll quickly walk you through these examples. Most of these examples are the exact same ones that sir also used in his videos, but I've just rearranged them so that this may be a little simple for you to understand, because now you have an idea of both the base functions in R as well as certain specialized libraries that require the use of ggplot. Okay. So, all right. So like we know that like I said, if I highlight a line, so this part, as we know, this is the R script, and here this is our console. So if I highlight this and hit command enter or control enter or alt enter, it's going to effectively get copy pasted below in the console and pressed and run. So it's going to be executed. So I don't necessarily need to highlight the whole or select the whole line. I can only press, like, have my cursor on this line and press control enter. So it's going to run and then give an appropriate output. So in this case, when I run this, these are the data sets which are available. For today's session, we are using a lot of these inbuilt data sets and a few of the same CSV files that Sir also used. So you can download each of them. So you might have already downloaded this, and or otherwise to practice, you can always download them from Sir's GitHub repo or the NPTEL website. Okay, so now scatter plot. So scatter plot is simple X by Y plot, right? It can have 4 quadrants, 1, 2, 3, 4, and these are basically dots. So that's what is known as a scatter plot. So if I just take this data, DNAs, it is available in R, but we don't see it in our environment here. So when I use this line, it gets saved into by the name D. And this is something that you might have seen in these lectures. So here, for some concentration, this is the density or the absorbance values that we get. So whenever you have 2 values and it's a paired value, one for the other, it's very obvious that an XY plot, an XY scatter plot can help us. So it's a simple plot function, plot, and the syntax is, that means the order in which you have to write. So whenever you're using a new function, it's always advisable that below in the console, you write a question mark, you write the name of the function followed by this curved bracket, because the function is always followed by the curved bracket, and you just press enter. So it's going to give you, if more than one options are present, then such a thing will come, but mostly we know what we are doing is, so it would be the same in this case. Let's just click one of them. So generic XY plotting. So the first argument is going to be what your x-axis is. The second argument is going to be your y-axis, and there are other arguments which are type. So what kind of a plot do you want? Main means the title, sub is like a subtitle, X label, Y label, and so on. So you will see throughout today's session that a lot of these plots, each plot has its own kind of arguments. You may be doing the same thing. Some of them use main, while something else uses title. So it can get confusing at first when you are just introduced to all of these new functions. But the best way to work is to keep practicing and then always taking help of this question mark function here. So, okay, so I specify the x-axis, I specify the y-axis, what my values are going to be. I also specify what my X label is, Y label. This is going to be specified in double quotes. PCH is, we'll see what PCH does. It's basically the design of the dot or the plus sign. So your actual, what is getting plotted, what symbol do you want? Color is blue. CEX, axis, label, all of this indicates the size. So this is something that you can keep changing. You can just do a hit and run, and this will be clear in time. So just make sure that always label axis. You should always label the axis and everything, the symbols that you put, the axis ticks, which are these markings, the axis labels and title, if you have any, all of this should be legible. It should be visible. Ideally, all of them should be in the same style and same font size. So if I simply run this, it's plotting. You can see there's a stop sign here, which indicates that the tool is running. And then I can expand this. So again, so this is something you've already seen in the lecture here. And if this is not something we want to plot, so maybe we want to take average of each of these values. So the aggregate function says that I want to find the aggregate. I want to aggregate these based on this. So for the same concentration, I want to aggregate all the density values. And when I aggregate them, when I club them all together, what do I want to do? I want to find the mean of it. And from where? From the data set D. So again, this is the same function that sir had already used. So we run this and we get this here. So now we have concentration values, and against each concentration, we have the average density. So now what do? We plot concentration again. Now see here, it was D dollar concentration. Now it's D underscore aggregate dollar concentration. You just need to be very clear with what you're plotting. So here we have 8 values. Here we have another 8 values. Everything else remains the same. And I just plot this. So now the plot looks good. Another aspect that you can take care of is it's always good that each of your dots should be within the X and Y limits. So as we saw in the previous one, which we can go through using this arrow here. So
hello, ma'am.
Yes. Hello. Any question? No question? Okay. If there's any question, just let me know. All right. So yeah, so it's always good to have these your voice is breaking. I cannot hear you. Can you maybe type in the chat box? Yeah. So the first time you make the plot, it will not be possible for you to know already the X and Y limits always. So maybe you can first make the plot, and then if you see if any of the X and Y limits need to be changed, then you can change them using this argument here. X lim, you specify the lower and upper limit, and similarly Y lim, the lower and upper limits. So now if you see here, we look at the original data. So let's take a look if you want to make the same plot with a differently styled data. So I had the CSV file here and I read it as E. So this is it is actually the same data, but it is arranged in a different way, right? So we have the concentration and we have different absorbances, not one after the other, like not one below the other, but one after the other. So here what happens is, so if you compare this and this one, we can sort of call the first one as a long form, right? And this one can be called as a short form. So these two are interchangeable. We'll see how to interchange them also. And it is also possible to make our choice of image using either of the two ways. So just like we had the row means here, so we want to calculate, so now like we calculated the means there using the aggregate function. Here it's designed in a different way. So instead of doing the aggregate function, we can simply find the means or the average of each of these rows except the first column. So row means the function simply takes a data frame as the input. So if I would have entered the entire E, it would have considered this also to be an observation, but we know that this is not there. So instead of doing the entire E, if I just write, so I just want everything except the first column. I don't want the first column. I want column 2 to 5. So I want all the rows. So pay attention here. So what I'll do, I'll write, okay, I need all rows. So before the comma comes the constraint on the rows. So I need all rows, so I won't put any constraint. And after the comma, I'll write C 2 to 5. So let's see if I get what I wanted. Yes, I've got these observations. It's like a new data frame now. Now, can anybody tell me, is there another way I would have got this? Instead of writing, I want columns 2 to 5, what is another way of stating the same thing? Instead of saying select columns 2 to 5, this is what the statement is saying. Could I have also stated this in a different way? I can give you a hint if required. So the hint is that instead of asking for columns 2 to 5, I'm saying give me everything except column 1. that should also do the task. It's the same thing. So if I say, give me all the columns except column 1 and give me all the rows, what can I write instead of this? So E square brackets, and there's a comma. So what do I write before the comma? What do I write after the comma? The first statement said, give me columns 2 to 5. This is going to say, don't give me column 1, give me everything except that. So if I say, don't give me column 1. Okay, if I wanted just column 1, what would I have written? Yeah, this is one way. Okay, correct. Now, if I wanted only column 1, what would I have written? You can unmute yourself and speak.
With exclamation mark.
Good thought. Very good thought. Okay, I like the thought process. All right. So exclamation sign is often accompanied by an is equal to or a true false statement. So here we are talking about indices, right? So if I wanted column 1, I would have written 1. If I said except column 1, I'll just write -1. So subtract that column and I got the same thing, right? So this is just an extra information. So there are again, doing this, there are multiple ways of doing the same thing. You're getting the same output. So coming back to why we did this in the first place. So, right, so we wanted to make a plot similar to this, right? So now I have this. So like we discussed the last time, whenever you have more than one function or something within something, which is known as nested, you always start from the inside. So inside here, it's this, which is the same as what we just wrote below here. And now I want to find the row means. So row means simply means that you sum all the entries in a particular row and you find the average of it and do so for each of the rows. So if I run this, let me just run this here. So I had 8 rows and I get 8 values. And since I want to plot this, I'll just save it in a variable. Now you can see that this is saved. This is not a data frame. A data frame would come up here. This is a list. So that is why it comes here below. And now what I want is, so what is the syntax of the plot function? The first argument is going to be the values on the x-axis, and the second argument is going to be values on the y-axis. They can be coming from 2 different places as long as they are paired. The first value in the first argument and the first value in the second argument, they would make you the first plot, the first dot, and so on and so forth. So here what I'm doing is, my concentrations are given in the first column. I did not add a column here which has my row means. I could have added that, or I could have it separately here, because I know they are in the same order. The order is not jumbled up, right? So I know that my x values are here as they were, and this is the y value. So I just need to specify them in the right order. So if I run this, now you see, this is the new plot. Okay, the values were slightly different than the old ones. So you can see these dots shifting a little bit, but you get a new plot which completely resembles the last one, which means that irrespective of the data being in the long form or the short form, we can make a scatter plot, right? So what do I have different from this line? So now this is a scatter plot, typical dot plot. Now, if you have used Excel, you might have seen cases where sometimes the dots are connected to one another. There is no fitting done, it's just connected. So instead of a dot plot or a scatter plot, it's now a dot plus line plot. So here type, so if I just go to this thing here, now you'd see type. So by default, it's points, P. That's why we did not specify anything. There's always a default value. Now, if I would have written L, I would have got only line. So let me just write L here and I'll write the line width. LWD is for line width. All the other arguments remain the same. So if I run this, I just get lines. I no longer get those points, but I know that this is not correct because I had points to begin with. So I'll write B, which is both. So I get my points as well as I get lines. So you can go through each of your old plots using this arrows unless you clear them, right? Now, this was discussed in the first lecture of this week. In the second last lecture of this week, or I think the last lecture, the last two lectures were also discussing about ggplot, right? So have you been able to install ggplot? Please let me know if, so this is actually ggplot2, right? Everybody else? This was not correct. Yeah, okay, all right. A lot of you have said yes, so that's great. Okay, so yeah, so you could have installed it from the tools here, right? Install packages, or you can also write this command, which is install.packages bracket, and in double quotes, you can write ggplot. And each time you are running this, so before running the command, every time you've opened a new RStudio window, you can just write library ggplot. So we'll do the same thing with ggplot also. We'll make the same scatter plot here. All right, so yeah, so I have a slightly different data for ggplot here. We can also actually make the same file. So wait, let's try with the same file here, the one that we used, this one. So now the only difference that we are going to do is, so here we calculated the row means, right? Now, can anybody tell me if I want to enter these row means as a new column in the same data frame? So let's say I have 5 columns here. I want a new column, which you can give the name. Can I, how do I write that? I want to save these instead of a separate list. I want to save it in the data frame itself. How do I access a column of a data frame? What's the symbol?
Dollar.
Dollar, correct. So I have my data frame, which is E. I write dollar. Now you see that I want to add a new column. So the column is not there, right? So I can write a new name here. So let's say I want to just write, let me just write row means, just for clarity. And then I would put the assignment symbol, and then I'll actually just save this because I already have saved it here. So I'm just assigning this value here. So now if I open E, you can see that this new column is here. So now I can actually replace this or not, does not matter. But now I'll use the same one to actually make the same plot using ggplot. So the syntax of ggplot goes that you first write ggplot. It's a function, so you write these round brackets and you write the name of the data frame. Here the data frame is E. Then comma, and I write AES, which stands for aesthetics, where I specify my X and Y axis. So my X axis here, I know my X axis is going to be so it's the name of the column without any double quotes. So here I write CONC, and then I write the Y aesthetic, the Y axis, sorry, without the double quotes. So here I'm going to write it as, I hope this was the spelling, row means. Okay, right? And now if I just write this, if I just enter this much, could not find the function. So I did not call the library. So remember the library's name is ggplot2, but the function's name is ggplot. So now if I run this, so what comes up here is, it read my data. It said, okay, concentration, I have these values which range from something like 0 to 12. And I have the row means, whatever it is, it ranges from 0 to maybe somewhere 2. But I don't know what kind of plot to make. Do you want a scatter plot, line plot, what color, what shape? We don't know. So I'll say, okay, you make a, so here it's called as a geom point. There it was just plot. Here it's ggplots, geom point. And if I just add this and I run this, now it's plotted. So, you know, it's the same plot, right? Same data, everything is same. Instead of the default plot function, we've used ggplot. Now we can make a lot of changes here, because you can see that row means is obviously not what you want as the name of the y-axis or concentration here. And do you want these grid lines? Do you want the dots to be black in color and so on? So I already have a few few data set that you would like to use below. So I'll just read this. So instead of the data that we were just looking at, now we have the same concentrations, but for each concentration, we have the absorbance values for an untreated sample and for a treated sample, right? So again, so we can just add, right? So if I run this, it's, so what it's doing is, so conc, and here I've just put treated, right? Because I have two columns. So I've just selected the one that is treated for the timing, and it's plotted concentration on the x-axis and these treated values on the y-axis. Now, if I also want to make the line plot along with this, I'll write this one. So in addition to these points, it has also made lines. And the final one, so instead of in addition to the points, I would like to add a smooth line to it, which is a trend line. So once I've added that, so you can see this is what it has done. So it was not a data that could use, it was not suitable for fitting. So nevertheless, it added this trend line, right? So you can see that this is how you build a ggplot. Now this is without any other changes, right? We have not specified, we've not beautified the plot, no legend, uh, labels are not correct, no units written. But anyway, the function actually becomes quite long. So you can see, as you will see below in today's session, that ggplot generally the function itself, the command that you would write to plot is quite long. So it's up to you. So one preference is that you write everything in one go, or the other thing is you build the function. So first you write just the basics. Okay, so I want to make a point plot or a scatter plot, and before plotting I'll just save it in the variable p1. No new plot has been plotted, but it just got saved. And then I say, okay, in addition to this plot, also add a line. So I say save it as p2, right? So now I can either print p1 or I can print p2. So these are two different plots. They're both there. You can use whichever of the two, right? So now you remember that in ggplot, there are only two aesthetics we are giving, x and y. In no place are we specifying two y aesthetics. But here we would actually like to make a plot where we have on the x-axis, we have concentration, and the y-axis should have both treated and untreated values. So this data frame is not suitable for use into ggplot directly. So what we are going to do is, so there's another package called reshape2. We just call the library, and it has a function melt. So what it does is it converts the short form into a long form. And the syntax is you give it the input E, and I say that keep the concentration column unchanged. So this column will stay, but I want to make changes into these columns, and these values are going to get shifted. So my ID is concentration, and the other two columns are the measure variables. So when it does this, so what it does, E new, it says, okay, concentration, and this was column 2, which became just a variable name here. And these were the corresponding values in that column. And this was the next column and the corresponding values in it. So I can just change the column names here. So I would like to keep them concentration type, which is untreated and treated, and absorbance, right? So if I change this, so yeah, these are my new column names. I can just take a look at this. And now this data is suitable to be used in ggplot. Now, if I use it just directly, let me, so now if you don't pay attention at the second column, if I just input first column and third column as my x and y respectively, so this is what it's going to plot. So you can see, you can very much see that this is one of the treated or untreated, and this is the other one. Now I want to make a difference. So I would want one of them to be in one color and the other one to be in another color. So in aesthetics, I write this additional argument, which is call. So color. So assign colors based on the second column here, which is type. So aesthetics, the inside aesthetics, the arguments will always be column names from this data frame. All right. So if I plot this, this is what it makes. It is randomly assigning one color to each of our categories. So there are a lot of other things that you can change here. So let's say I want to increase the size. So I'll just add this. So the size is also change the size of these dots. And if I want to make a smooth line rather than adding a just a line, I would actually like to add a smooth line to it. So here I'm building on this one. So G1, and then I add this thing here. So it adds these smooth lines. And furthermore, on this plot, I would like to add these labels. So on the X label, instead of CONC, I would like to add conc with its units, absorbance with a capital A, and instead of this, so here I said color, right? So my legend effectively is what's coming from this. So I'll just write color is equal to sample type. And I would also like to increase the while lim here. So I run this, and these changes have been made. And a lot of times this grids don't help. So classic theme or the theme black and white are most used. So this is ready. So G4 is also this. And if I did not have anything, but I can also make a new plot directly. So it's the same thing. So I think the only difference was size. So I can increase the size here. And it's that same plot again, right? So it's up to you. It's your personal preference whether you want to build on ggplots or you want to write them all together. It's absolutely your choice. Any doubt till now? We are done with scatter plot for the time being. We will go on to the next type of plots. All right. Any doubt until now? It is always very helpful to understand each of the arguments that these functions take. And if you get stuck anywhere, you can always Google these things that, okay, I want to add color, I want to change color of the plot. So where exactly would this go? And by what argument I can access this feature? But until then, if you have any doubt, please feel free to ask me. These doubt sessions are specifically meant for that. All right. So if there are no doubts, then great. I'll move on to the next topic, which is the bar plot, right? So not all data is suitable for all kinds of plots. So I'll remove these for the time being, and we'll read on this new data, which is this one. So this is the same as what was used in the lectures. So I would just like to know the distribution of my cells in the different cell cycle phases when it's the control condition, or it's the first kind of drug or the other kind of drug, right? So by default, a bar plot takes a data matrix, right? So can anybody help me recall what is the difference between a data frame and a matrix? The major difference between the two? I hope you're still there and not sleeping. What's the difference between a data frame and a matrix in R? You can write it in the chat. You can unmute yourselves. If you don't remember, let me know that too. No. Okay. No issues. So
Data
a
frame will be like tables,
yes, and a matrix.
like rows and columns.
So 2D, you mean, right?
Yes.
It has rows and columns. So matrix also has rows and columns. Correct. So a matrix will always have one type of data. All the rows, all the columns, all the elements, if they have numbers, all of them will have numbers only. And if it has characters, that is alphabets, it can only have alphabets. On the other hand, a data frame, each column can be a different data type, right? So if you can see the data frame that we began with. So by the way, if you want to see the data type of anything, we can use this structure or the class function, and right, so it says it's a data frame. So if I want to convert it to a data matrix, let's say this is, so it converts it to a matrix, I can just enter this. So what it does is it does something here, right? Can you see the first column? This is not what the first column was, right? It was something else. It's randomly assigned numbers to it. I think it assigned it alphabetically. So drug A came alphabetically first. It was 1, 2, and untreated came the last. So we know that, so it is coercing. It is changing the data type from one to other forcefully. But this is not what we want. And anyway, we know that when we are plotting something, we would not need this information as it is in the plot. So we would only take this. Now, just like I asked you a few minutes back, I want to get columns 2 to 5, or I want to remove column 1. So how do I do it? This is one way, but I don't want to write more. So what can I write here?
-1.
Right. So, sorry. So here, if I just want, so if I want the whole thing, so this is what it's going to be, f all 2, and I can, so just to wait, before that, I'll just write the control one. So simply, so a bar plot is, so if I see what the matrix looks like, so it has these 4 columns. The column names stay intact, and I have these values with me, right? So I just want to plot. So 278 would be a bigger bar, 10 and 10, and so on. So if I just write bar plot and these 4 values, it's plotting them. It's taking these column names as the x-axis by default. No formatting required. Now I know these are percentages, so I would like my y-axis to be from 0 to 100, and I can just write, so it would be percentage of cells, right? So this is how my final plot should look like. Now I want to plot it for all my all the types of samples, the control and drug 1 and drug 2. So I have this. So if right, if I just write this, right? So beside I need to write, because there are multiple values here. F1, okay, this should be 2. So if I write this by default, right? So I just say that I have each of these values, and you set 1, 2, like this 2, 3, and 20, just set them one beside the other. Then 78, 80, 54. And then 10, 8, 14, and then 10, 9, 12. One beside the other. So which is why I wrote beside here. Now let's actually try if I remove beside what it makes. So it made a stacked bar plot. And you can see now these values go well beyond 100. But in our case, we know we are just dealing with percentages. So the stacked percentage don't make sense. In some cases, stacked bar plots can help. So in that case, you're effectively removing beside, which means you're writing beside is equal to false. So, but in this case, we'll be writing beside is equal to true. We know the y limits, we know what the label should be. And now another thing is, let's say I would have written beside is equal to true. Now, I don't know what my first one here is, second and the third one. I don't have a legend for these colors. So from the original plot, from the original data frame that I had, I know the first one is untreated, the second is drug A, and the third is drug B. This order I know. So this order I can access how I want to access this column basically. So I'll just write F dollar, the column X. So I have these things in these value in the order. So what I've written is that legend text should be this 3 in this order. That's all. And this one is just specifying that I want it on the top right of my plot. And this thing is up to you if you want to change the color of these plots. And this is just increasing the size of all the labels and the ticks. So if I run this, everything's set. All right. So this is bar plot using the base R function. This is not using ggplot. So this is using the base function bar plot. Next, we go on to making bar plots using ggplot. So for simplicity, I have chosen a different data set, the one that was also used in the lecture, the one that we also used last time, the iris data set. So now we want to make a bar plot, let's say, where our x-axis is going to be species, and we just want to find the petal length here. So now here I know that, okay, I think before that we can again go for the same data here. So let's say, so I had this F plot, right? So, okay, so let's say I actually want to plot each of these plots. I think we can come on to it a little later. Right, just remind me at the end, if time permits, we will make a bar plot from the same function using ggplot. So before that, this is what was discussed in the lecture. So we can simply make a bar plot. We just need to specify the x-axis and we want to specify the y-axis. But now we know that bar plot is just a single value per value on the x-axis, right? So here, if I want to say, let's say I want to find the petal length for the Setosa species, I have so many of these values. I can't plot everything as a bar plot. So what I'll say, I'll take all the values, find the mean of them. Sorry, right? So I can find the mean of this and make a bar plot. So this makes the bar plot for each of the species, the distribution of petal length. And now typically we have calculated the mean, so there's also going to be an associated error bar. So this is the same error bar, stat summary function that's calculating errors. So if I run this is the plot. And so we have just separated this. So make a ggplot, on it add a bar plot, on it add the error bars. Now if I can just alter the order, so if I say make a plot, you already have calculated error bars, add the error bars, and then actually make the bar plot. So then it looks more cleaner, right? And then I can make some more cosmetic changes here using adding the labels, using the classic theme. And here also you can see that once I have used the theme classic, and then I've overwritten on it to add these two. So if I run this here, this is what I get. Now, just like we discussed that the order is important. So let's say I would have had the classic theme after. So you see, none of these changes are actually taking place, right? So the label size has actually reduced. You can see the difference. So the order of your arguments is sometimes important. In ggplot, especially, you need to take care if any of the two arguments they overwrite one or the other, then which one should come first and which one comes after it. All right. So this is something we would not be using. Okay. All right. So yeah, so I just did this comment, but now this is a multi-line command, right? So not all the lines they got commented. So if I want to add a multi-line comment, I'll write Control-Shift-C. All the lines get commented. Okay. Any question about the bar plots using either of the two types of functions? If not, then we will go on to the next kind of plot, which is a histogram. Yeah. No doubts. Please ask if there's any doubt at all. Yeah. That's all. Okay. Okay, fine. So next we move on to making histograms. I'll just clear this plot here. Okay. So for histogram, again, the same data set that was used in the lectures. So this is a flow cytometry output. And so we can make a histogram of any of these columns, right? Let me close these as well. So let me show you a histogram. Let's try making a histogram of the first one, fsc.h. Okay. So I'll write, so the base function is hist and f dollar. I get this. So the first one, and I plot this. This is the distribution, right? I can do it for, sorry, f dollar, second one. So any of these is possible, okay? And once you know what you're plotting, so here for the example, I have used f dollar fl2h. So here this is the distribution. You can, since you know that again, this could be somewhere around 4,000 or so, and maybe this is extending beyond 800. So you can just set the x and y limits. You can write the x and y labels. If you don't want this header here, you can just write main is equal to double quotes with nothing. So it's an empty header. What does LAS stand for? I'll just write hist. And let's search where LAS is. I think this is something. Let's try running this. Yeah, okay. No difference. I would have added LAS is equal to 1. Okay. You can see this difference, right? So in the previous one, it's just skipping these labels. So I said that label all these values, right? Okay. Now, okay. So this is the default, right? So it decided how many bins to make, and it counted values in each bin, right? This is how a histogram is plotted. You have the distribution, you had values from 0 to 1000. You said, okay, let me make bins of, let's start with 50. 50 looks good. So 0 to 50, I count how many values are there, and I have the frequency now. So I said, okay, maybe 100 values lie within 0 and 50. Then I see 51 to 100 or greater than 50 to 100, and I say, okay, 1000 values lie in it. So this is what a histogram is doing at the back end. Now I would say, instead of these breaks, instead of this number of breaks, you make 10 breaks. So if I ask it to do that, it's making 10 breaks. So that means it's making breaks of 100 each. So there are two things. One is number of breaks, and one is the width of each break. So here in the histogram function, it's using the, it has the argument breaks, because one is same as the other, right? So here we are just talking about breaks. So number of bins, that is. And so this Friedman-Deconis rule, this is something that can give us a better distribution, the best number of breaks, so that you don't have to do it using a hit and trial method. So this is a histogram. And if you remember from the last lecture, the week 3's lectures, so abline is a function which is not making a plot, but it's just making a line on an existing plot. So what we say here is that you make a line, we want to make a vertical line, which is actually the mean value of our distribution here. We had all these values here. I just want to find the mean and plot that as a vertical line. I also want to make another vertical line, which would be red in color. That would be the median of these values. So it's not making a plot by itself. It's making lines on an existing plot, right? So just to show what that means is, if I just clear the plots, so it says, yeah, okay. If I just run this, it says error, new plot has not been called yet. So there is nothing, no plot here to make these lines on. So I have this plot, and then when I run this, I get the first line, I run it again, I get the line for the median as well. Now, this was something we discussed. So by default, there are two options present in the histogram function. Either you can plot the actual counts or the frequencies, or you can plot the density. So density means if the total number would have been 1, what would have been the density at each point. each of these bins, right? So, yeah, so by default, this is what we plotted, counts, right? Now, if I write this argument, frequency is equal to false. So by default, it takes density. You can see this is taking density, right? Because now you can see how that works. If you observe, yeah, so the distribution is not changing. It's just the y-axis that is changing. And that is what we expect. But now, instead of density, I want the percentage. So in terms of 100, right? Out of 100, how much percentage is this? So what I'll do is, instead of plotting, we save so just like in ggplot, we can save that plot to a variable. Here also, we are saving the plot to a variable, temp in this case. And temp will have breaks, counts, density, mids, xname, equidist. So these two are of our interest. We can plot counts when the frequency argument is not given, and we can plot density when the frequency argument is given as false. So we will tweak this a little bit, and what we'll say is, so when we say frequency as a percentage, so percentage is values of your interest divided by the total number of values into 100, right? That's the formula for percentage. So if I just write, percentage is what? Percentage is your values into 100, and then you divide it by the sum, or like the total value. So here what we do is, for counts in each bin, what we do is take the value, multiply it by 100, and divide it by the sum of all this. So all the observations that I have. And now I cannot make a new variable in the histogram. It only takes, it either plot counts or it will either plot density. Density is not of my use here. So I will actually save this information in the density thing. So if you see here, the density right now is some 0.000 so on. And now if I save this, the density, okay. Wait. Yeah. So right. So now the density has actually changed. Let me reopen this. Right. So now you can see the density has changed. Right. And now we are back. So now we have plotted percentages. I hope this was clear. Please let me know if it's not. We'll move on to histogram using ggplots then. Right, so we'll make use of the same data F, the facts sorting. And right, so now what it does is, in histogram, there is no y-axis, because y-axis is basically counts or percentages, right? So we just specify the X aesthetic in this. So we just specify what column of our data frame we want to make as a histogram. So it's making this histogram, right? It looks very shabby at the moment. So there, if you remember, we had this thing called breaks. Breaks is specifying the number of bits. Now here, instead of breaks, there is an argument known as bin width. So you can specify the width of this bin. So if I specify 6, then it's going to be a shorter bin, right? And so on. And like anything else, I can add labels. I can change the Y limit. I can change the theme. I can add some other theme. And this is what it looks like. There are a lot more things that you can change, specifically if you want to make the border outline of each of these bins and you want to fill inside as a different color, as was the case here. You can make those changes, but that you can explore. All right. Everything is going to be here within the aesthetics. All right. So we run with the histogram also. So you can make a histogram with either of the two functions available. All right. As we decrease the bin width, the number of bins increase, right? Because let's say you have your values ranging from 0 to 1000. So I say that number of breaks, let's say I want 500. So if I write here, so number of breaks or number of bins multiplied by the bin width. This needs to be equal to our total observation, right? Our actual, the interval of the observation. So in this case, it is 1000. So if I say now the bin width, now let's say the initial bin width was 50. So it was 20 number of bins. So 50 into 20 is 1000. Now if I decrease the bin width to 5, so it's going to be 200, right? So this needs to be followed. Yeah, all right. All right, good observation. So with that, we come to box plots. So most welcome. All right. So with that, we come to box plots. And here we are going to use the insect spray data. So let's see what it looks like. So spray 1, how many insects were left after spray 1, after spray B, C, D, E, and F. So now a simple way to decide if a box plot is going to be a good fit is that you can imagine your categories across the x-axis always. So case 1 to case 2, case 3, and so on. And y-axis are going to be corresponding values. And for each case, if you have multiple values, then a box plot is the best suited plot in most cases. So in this case, since we have these categories A, B, C, D, E, F, and for each of these categories, we have at least 3 counts. So it's a good practice to make a box plot. So box plot, the way it's done is y-axis tilde x-axis, right? So here you have a box plot, and on the y-axis we want counts, and on the x-axis we want the spray type. So we have y tilde x. And from where are the columns coming? From the I data set, the data set that we have saved as variable I. And if I only run this much, I have the box plot with me, simple box plot, looks good. I can just make such changes. I can say that the outlier should be a circle, filled circle, red in color, y limit from 0 to 30. Variable width indicates a different width box plot depending on different number of samples. Here it's the same, so it would not make a difference. And again, this label size, access size, the font size of each of them. If I change it, same box plot, just the organization has a little changed, right? So like we just discussed that arguments, so box plot has these arguments. The simple plot has some arguments, some common, some different. Histogram has a different set of arguments altogether. Bar plot has different ones. So it can get confusing at first. So it's always good to look at the manual, the description of these functions, and then determine what argument goes where and what would you like to specify in each of them. And with practice that comes, it becomes easier. This is slightly simple when we talk about ggplot because there's a certain trend to all of this, right? So you'd see there's more uniformity of these arguments in ggplot, irrespective of what kind of plot you're making, right? There's always going to be an aesthetic. You will always write something here, and it's always going to be followed by labels and themes and limits and so on. So for ggplot, again, same thing. You specify the x. Here we have spray, the y thing, count. Color I can specify if I want it to be in a different color. So here I'm specifying the spray column again, which was my x-axis. I'm saying that, okay, you change colors according to different values that are there along my x-axis. So each of these box plots will become a different color because I have six unique values in my column that I give to the color argument. So if I just write this in ggplot, this is my plot, different colors. a legend also comes up. So I just need to write these. So I'll just make the same changes. Let me know if I need to explain these. So here I've specified show legend is false, because now you see A is orange. If I would have just hidden this part, I still understand that, okay, A is orange in color, B is yellow in color. This is not adding any information to my plot, so I can remove it. So I said show legend is equal to false. So I run this. This is fine. The legend was not adding any new information. If you wanted, you could add the legend, and then you can also add the label for the legend here as well. Right? So here, if it was color, here it should have been color is equal to, let's say, right, spray, and here it would have been true. If I run this, get this again, right? So either of the two things you can do. Simple box plot using ggplot, right? Any confusion? Any doubts? It's always a good practice to keep saving your R script. So you can see here this asterisk sign indicates that my R script is not saved. So in case my computer shuts down right now, I might lose my script. R Studio generally retains the last script, but it's never a good practice to rely on that. So always save this. All right. Okay. Next, we can begin with the heat map. So heat map, we are only using the default R function, which is heat map, not anything from ggplot here. So I have this. So let me try and open the exp.csv in Excel. So this is just a different thing. So it's always good that when you are reading a CSV file, you should always take a look at how R has read it, how the resulting data frame looks like. So this is what it was. It's a very big data set, but let's say this is what it looked like, right? So I had these columns here and then these row names, and each of these values correspond to something, which we'll just discuss what this is. Okay. So now I read the CSV file. This file looks normal, so I read it. So we just saw it's a pretty large file, which has some 12,000 rows, 12,000 observations, and 48,000 columns. So if I look at it now, I can see, yeah. So I can see that it has read everything, right? So typically you would see this is, if I tell you this is an RNA-seq data, after some analysis. So what it's basically showing you is that on the columns, these were some samples, some sample ID 1005, 3002, and so on. And why have they been preceded by an X? So R by default does not start a variable with a number. You can specify a variable as A1, but it would throw an error, or it would give you a warning if you write 1A. So it's always a good practice to have variables starting with an alphabet. So here it said it recognized this as something which was beginning with the number 1. So it just preceded with a capital X across all the columns. Okay. So here these are column names, right? Column names are my sample names. Normally what is suggested is that you should have these features. Let's say I call them the names of genes. So I want to call these as my row names. Right now the row names are 1, 2, 3, 4, 5. So, but since this thing is not adding any information, so otherwise it was all just numbers. So I would want this to actually go into the rows as row names. So my column names become my sample names, and these gene names become my row names. So a way to do this is, so what we can obviously do is, we can say that row names of this data frame, and you assign that from the first column. So let's actually give that a try. So I have this here. So I say row names of E, and what do I want this value? So here I want it to be E dollar, the first column, right? So if I run this, I'm assigning E dollar X as the row names for E. So now you can see that this is what has happened, right? Now it is redundant information. I have the same thing in two columns. So now I want to get rid of the first column. Now in this case, I have actually forgotten how many columns were there, 48. Okay. So, but I, again, so this is why initially we talked about the importance of writing -1. It's just simpler. You have you have less columns to remove than what are there. So to avoid any issues, what I'll write is that all columns, sorry, all rows and everything except the first column, and I save it back in E. So I'm overwriting. So if I save this, I'm removing the first column. So this is what I get. And this is what is sort of our expected data set, data like data frame type. This is what it should look like. So one option is that we do these 3 steps. The other option is that while reading the CSV file itself, I say that row names, so take the first column and assign it to row names. And don't, then naturally that column does not come up in my data frame, right? So if I run, let me call this E2 so that we can compare. So it's reading the file, it'll take a moment. It's read. So you see it looks the same, right? So let's see E2. That's also the same thing. E2, they're both the same. So either of the two ways you can write. So it's always recommended that these features, whether these are gene names, something to do with transcripts, something to do with proteins, whatever these are, these should be row names, and your sample information should be columns. All right. And now we know that each of this is now our actual data. Everything except the row names and the column names, everything that is a number. These are numbers. So now we can convert it into a matrix. So we convert this to a matrix. We have EMAT. If you look at it, these 3 are absolutely identical. So just to check if it's actually a data, whether it's a data frame or a matrix, we write E and we write E underscore mat. All right, no issues. Yeah. Okay. Yeah, in case any of you have any doubt, yes, the recording will be available if the session is being recorded, and I will upload it after like within an hour of completing the session. All right. No problem. This is why the sessions are recorded, because it's not always possible to devote 2 hours on a Monday evening for this. So you can always go back through these recorded sessions and see if that clarifies your doubts. Okay. All right. So fine, so we can see that the class, which means the data type, is a data frame in one case and it's a matrix in the other. All right. So why we converted it to a matrix was because heat map, this function recommends taking numbers. Now, what is a heat map? So if I take a look at this type of a matrix or a data frame in any format, it's known as a count matrix. So these are counts. Now counts typically are going to be integers, right? They're going to be, in fact, more than integers, these are going to be whole numbers, 0, 1, 2, 3, 4, and so on. Here they are decimals because they have been normalized through some way, which I would not be discussing at the moment. But these are numbers nevertheless. They indicate some sort of count. That means, let's say, let's just assume this was 7 for the time being. So in sample number 1, the counts, let's say the counts of RNAs of this gene were 7. And in the other sample, the counts were also 7, and so on. Mostly these were 7 in all of these cases, right? It's more or less the same, right? Or if we take a look at anything else here, right? So we can say here it was 5, here it's 6, and so on. So these are counts of a gene in some or the other sample. So here it's difficult to look at, right? So it's 7 in one, okay, it's almost 7 in the other, and you can't look at all of them in one go. So a heat map is just a graphical representation of a count matrix. It's as simple as that. So you have these values, and let's say the lowest value was 0, the highest value was maybe 10. Now you're just saying, okay, I'll assign one color to 0 and I'll assign one color to 10 and everything in between is going to be a gradient. So this is what a heat map is. So a heat map, the rows and columns are going to be the same. The rows are going to be your sample. The columns are going to be the genes in this case. So let's say I plot this. So can anybody tell me what I'm doing here? How many rows and how many columns am I taking? In terms of how many samples and how many genes am I taking here? 1 to 20, comma, blank. You can unmute yourself and just say. So I'm basically subsetting my, sorry.
1 to 20 rows and all columns.
Right. So 1 to 20 rows means that I am taking 1 to 20 genes, and all the columns means I'm taking all, how many do I have? I have 47 samples. So I'm taking all 47 samples. So all 47 samples should be my rows and these 20 should be my columns, right? So I'll make a heat map of it. Okay, made this heat map, right? Let me just zoom in and yeah. So if I just show you here, if the small plot, hello, I hope you can hear me still. Yeah. Yeah, I think the internet was just some internet issue. I'll wait for the others to join, right? Yeah, we'll just start in another couple of minutes. More people are rejoining. Yeah, in case such a thing happens, you can always rejoin within a couple of minutes. I think that should be manageable. Do you have any confusion by the time everybody else is joining? Want me to clarify anything else? Okay. All right. So just give me a minute. Okay, I'll share my screen again. All right. So this is where we were, right? So here you can just see that, so right, my rows are the sample names and the columns are the genes. Now you see here, I had taken 20 genes here, but not all 20 genes are labeled. Here I had 47 samples, but these names are, the font size is just larger. So if I zoom it, then everything fits, right? So this is something, this is a both, it's actually a nice feature of R that once you zoom it, you can actually, you can actually resize it if you resize your plot. So it can be good in a lot of cases. So just be careful that when you are plotting, you can, is it, if the font sizes and everything, if the proportions, they are fine. Because you wouldn't, so a heat map like this, so if it's a 20 versus 47, do you want it in a square shape? Should it be rectangular? Ideally, it should be rectangular because 20 by 47, right? It's not a square. So either way, you can if you want to change the font size, like let's say in this case you want, so the default size is 1, so I want to, let's say, half it. So I'll just half these values, so now everything is fitting. Okay, now there is a concept known as scaling. So what happens here is, so let's talk about so let's say the first, so here the colors are, yellow indicates a light color, and let's say this dark brown indicates a the higher values. But now the thing is, the highest value, so if I see, let's just talk about these 4 values here. Okay, so let's say 7.59 was the highest value for my in my first sample. So okay, before that, when I'm reading a count matrix, does it make sense to read it column by column, or should I read it row by row? That means, should I consider all the patients, and I should, let's say, focus at gene 1 and look at that in all the patients, or should I look at the first patient, or whatever, the first sample, and look at all the different genes? So typically it's read as per the column. So the first patient, the first sample, it has all the genes. The second sample, all the genes, right? Like that. One gene never comes together in all the samples, right? That does not make sense. So I would like to see the distribution, or the distribution of different genes per sample, right? So let's say I look at the first sample here. So this thing, first column, and I say, okay, 7.59 from the first gene, that's a higher value, all right. 5.0, it's a lower value. But how lower is the value? Were these like the top 2 values for this sample? Were these like the lowest, most 2 values? So 7.59 here and 7.56 here, they are actually different. I cannot compare these two, but these comparisons are more meaningful. So in addition to whatever transformation has already been done on this data, there's one thing known as scaling. So scaling, if you remember, scaling means centering the data around 0. Normally it's centered around 0. So what I do here is, I'll just scale it column wise. I can also scale it row wise, but in this case, it does not make sense physically. So if I scale it row wise, you can see this. So now it means that this gene has a high value in this sample. It was relatively high. So in this sample, all these genes had a higher value with respect to these genes, right? So again, all these genes here, they had a higher value in this sample as compared to this. So I can compare it within a single column. So I have these categories. So now I can say these genes were the most expressed genes if they are talking about RNA molecules. These genes probably had lesser RNA molecules, and these genes had really low RNA molecules. So the scaling helps. All right. And the last thing is these dendrograms here. So if you know the concept of phylogenetics, this is just showing how similar are any two things. So here in this case means that these two genes, they behave quite similar, because if you see these two genes, they have a similar expression profile across all my samples. These two, they differ a little bit, but still they're quite similar to each other. On the other hand, this gene versus this gene, they're quite different in their expression profiles across all the samples. So this dendrogram is about genes, right? And then this dendrogram is about samples. So that these two samples, they behave quite similar, right? Or maybe all of these have a similar expression profile. And then maybe this clade, you see, this is quite similar. So these patients have a similar profile. But a lot of times, dendrogram, so it depends on what you're looking at. Do you want to look at similar gene sets, or you want to look at similar patients that are behaving similarly? So we might want to remove the dendrogram in some way. So whatever clustering you want to remove, you just specify that as NA. If I want to remove this clustering, these are columns. So I'll write col V as NA. So you see this has gotten removed. right? And I think I need to add this again, right? So if you see this one, so the genes have been rearranged, but the order in which your patients occur, they are the same as was in your initial sample. So X1005 was the first one. Yeah, 3002 should have been the next one. Maybe it's not plotting all these values. We can extend this. Yeah, okay. I'll have to recheck why it's not plotting 3002 again. Yeah, it's plotting it here. So it was not able to plot those. You can see this order is exactly same as this order, but this is not the same, right? So 1080 was the first gene, but 1080 is not the first gene here, right? It's somewhere in the middle here. So genes have been rearranged based on their similar profile across samples, but patients have not been because we removed that clustering. All right. And I can also, if I remove the row clustering also, so what happens is, now you see, so here you can say that, okay, maybe this gene 10980 and all the ones that are in deep red, they have very similar profiles. The ones in orange, they have another kind of profile. The ones in light white and yellowish, they have a different profile. So I should actually have them together. I can make some conclusions. Are those genes, do they have some similar function or something, right? So this is, you need to think where clustering makes sense. Do you want to cluster along the samples, along genes, anything? So heat map is a slightly difficult concept to grasp at first, but just remember that this is a graphical representation of your count matrix, nothing else. Okay. I think this is the last thing for today, volcano plot, right? And then we'll have some practice questions to solve. So if there are no other questions about anything that we've discussed till now, I'll quickly go through the volcano plot. So we just talked about RNA-seq data, right? In the data that we've just currently plotted. So if you take a look at, let's say, first gene, so let's just say half of these were healthy people and the other half were people who were suffering from some disease. So we have just done an RNA-seq. So we're just looking at the distribution of RNA molecules in the blood cells of these, let's say 20 versus 27 samples. So now I see how many genes are there that have a different distribution in one group versus the other. That means, let's say there was a gene A, it had values about 5, 6, 7, 4, 4.5 in one group, and the other group had 20, 25, 10, 15, 22, and so on. So you can make a box plot, right? You can imagine it as a box plot. Controls and the other group. So one has something around 5, the mean is around 5, and the other, maybe the mean is around 20. So you can say this and these values, they have a change. This is known as a fold change. How many times the values have increased or decreased. So a fold change of 2 means it has doubled. Fold change of 5, it means it has multiplied by 5, and so on. So typically these values are also, instead of saying 5 and 10, they are set in terms of log 2. So log 2, if the value had changed by 2, so the log 2 would be 1. If the value has halved, log 2 would be 0.5. Log 2 would be -1 if the value was 0.5, right? So this is known as log 2 fold change. You're comparing this and this, and you're doing a statistical test to test if this is an actual significant change. So you get an associated p-value. So this is what is known as differential gene expression output. This is what we are going to look at here. So I read this. So this is a gene, and we say that the log 2 fold change of this gene was -1.2. So it means the value had reduced, and the p-value is something to the power -34, and so on. So these are all the genes. I think they are arranged based on their increasing p-values. So you can see the values here. You have a non-significant p-value. Typically, again, cutoff, you can take 0.05 or you can take 0.01. So this is what we have. So now there are two values here. You have the log2 fold changes and you have p-values. When you have two values, they are paired, what kind of data fits the best? A scatter plot. Something on the x-axis, something on the y-axis. So let's just make this plot, right? So I'll use the simple plot function. And let's say I would like to have log2 fold change on the x-axis, and I would like to have the p-value on the y-axis. Let's just say for starters, I want that. And I plot this. This is what I get. Now, so I know my p-values range from 0 to 1. So this is what it is. And I know the log2 fold changes, they can be greater than 0, they can be less than 0, so they are here. Now, this plot, while it is informative, it's not very informative. So people have come up with a slightly different variant of the volcano plot, which is known as the volcano plot, where they are plotting almost the same thing. But what they're doing is, so again, we'll just use the scatter plot. But instead of plotting p-value, they'll plot the minus log 10 of p-value. So why it helps is, so what I can do is, instead of this, so log 10 is a function, you remember? Right? Log 10 is a function. So I'll write p-value on this, and we know p-value is decimal. So these log 10 changes would going to be minus. So I'll just put a minus sign before this so that I have positive values. So higher value of minus log 10 p-value would indicate a lower p-value, which means more significant. So I'll just write this here and I'll make this plot. P-value not found. Sorry. So this is going to be V dollar p-value. Now you see, this is what we get. This is a volcano plot. So why it's simpler to understand is that normally we take a cutoff that anything less than -1 log2 fold change makes sense. Anything greater than 1 log2 fold change also makes sense, given that their p-value should be above a certain number. So above what number? Again, p-value we take we take 0.05. So we'll do the minus log 10 of that. And what would that be? That would be 1.3. So anything greater than 1.3 and fitting in these categories is a good. These genes are of our interest. So doing the same scatter plot using ggplot, the same plot we get. Now there you remember we could add AB line, and we can write the x and y intercepts. Here also it's, instead of AB line, it's geom V line, which means a vertical line, geom H line, which means a horizontal line. And we run this and we get this. So these are the genes of our interest. These are the genes that are of our interest, which have actually have a significant p-value, and the log2 fold changes are also either less than -1 or greater than +1. So if I say that my log, so here it was V dollar log2 fold change, this should either be greater than 1 or it should be less than -1, right? So another way of writing this is, if I use absolute. So ABS is a function, which means absolute value, which means remove the sign, make everything positive. So V dollar log2 fold change. Now I can say this should be greater than 1, right? So either you can have these two categories or you can have the same single category, right? So this is just for your information. So we are interested in those cases where the absolute log2 fold change is greater than 1 and where the p-value is less than 0.05. So we have plotted this. Now we just want to make a few more colors here. I want to color this, the ones which are down expressed or down regulated or under expressed. They are normally shown with maybe red or green color, and the other one with the other color. So it's one of the two colors that you can use. So what I will do here is, so this was my original data. So I am going to add another column where I would write the information, what is the direction of fold change. So if it's less than 0, if it's less than -1 and the p-value is satisfied, then maybe you write down, otherwise you write up, and so on. So how I'm going to write this is, I want to add a new column. So the best way to add a new column is just specify a default value. So I know most of my genes don't fall in any of the two interesting categories. So I'll just write no. So make a new column and just fill it with no, like this. It's a new column, diff-exp, and it's just no all throughout. And now I change some of these values. So what I do is, so you remember this conditional statements that we talked about? V dollar this greater than equal to 1 and V dollar p value should be less than equal to 0.5. If this is satisfied, you write up. Now, so if I write, so why is this? I'll just walk you through how it's written. So V dollar diff. What do you think is the dimensions of V dollar diff exp? How many, what values are there and how many values are there? Is it a data frame? Is it a matrix? Is it a list? Is it a single value? Just this thing. These are the last few questions I have for you for the day. I'm taking one column from my data frame. So what is the dimensions? What is the type of information in this? Come on, answer. It's a vector, very nice. So it's a list, right? It's an array or a vector or a list. Now it's one dimensional, right? It just has values one after the other. So, so that means if I want to access elements, I would just write a square bracket. And if I write 1, I'll get the first element. If I write 2, I'll get the next element and so on. So then what I'm doing is I am using a different column altogether. So V dollar log to 14, this is a different column. Now this, if I write only this statement, so what is going to see is it's going to take each of the elements of V dollar log to fold chain and it's going to see what are they greater than equal to 1 or not. So if I write this and I enter this, I get a list of true, false, true, false, true, false. And if you see the length is going to be equal to 12625. Likewise, I have this. I apply a constant on the p value column. If I write this, I'll again get a list of 12625 true, false. And if I do AND, I'm using that AND gate, right? Relational operator. Sorry, this one. I again get a list of true, false, but now it's pairwise, right? If both of them were true, then it's true. If either of them were false, then it returns false. Now I feed this into this. So effectively, I am feeding, inside this, I'm feeding a list of true, false, true, false, true, false. So whichever were true, those values I'm going to get. So I'm saying that wherever this is true, you assign it as up. So wherever these conditions are satisfied, wherever the index here comes out as true, you assign it as up because they were upregulated. And if this was less than -1, but the p-value is also satisfied, I write down. So now you see some of these are down, no. I think log to fold change, you would see a lot of them. Anyway, there would be some more which are going to be in the up category here. Right? So now what I write, I just want to make changes into the color. So I want to get 3 colors now, one for up, one for down, one for no. So if I just run these 3, right? If I just run this much, then I get these 3 values. So by default, ggplot has said, okay, I'll assign red to the first category, green to the other, blue to the other. If I want to assign the colors of my interest, I want this one to be actually black because they are not of my interest, right? These are not differentially expressed. So I'll just say red, black, and green. And if I run this here, red, black, and green, and I also specify clean theme classic, and that's it, right? So normally they are assigned alphabetically. So down, it comes first D, and then N, no, and then up as U, right? Nevertheless, this is what I get. So that's how we've made the um, volcano plot. Now, one final thing remains is I would actually like to know what gene this was. I don't want to go back and look, okay, uh, there were so and so. So this has approximately minus 1.2 and p-value was, I calculate it back. I back calculate to find what the p-value was. It's going to be tough. So I'll just add labels here. Now I know that the labels are already there at this first column, but I don't want to plot all the labels because I don't want to plot these. This is going to be a mess. So what I do is I make another column. So like we did before, I'll make a column and it would be null, or you can just write it as empty, right? So if I add this, it's just an empty column. There's nothing in this column. I've called it lab. And then I say, if we just saw that if differential expression, if here I have either I have up or down, that means I do not have no, then you assign that value from the gene symbol column. So you transfer the value from here to here, provided this is down or up, not no. So how that works is, right? Okay. Warning symbol. Why does this give this change? Let me just see. Okay. Nevertheless. Okay. So probably what this means is that some of our last observations just give me one more minute. I want to see why we have got this. Thing is saying, so it should not be a problem. It's a warning, but we just check that this, and this, each of them have the same length. So this, I'll check why the warning has come up, but I think we are good to go. So now what differences I add? I'll just write label is equal to my lab column, and that's it. So once I add that, I get these labels. So now I have the gene names. So I know that these are the 5 genes that are down-regulated, that are up-regulated, that I can work with ahead. So with that, we come to an end of this part. Now I just have a few questions to quickly brush up. So just as a practice for this week's assignment. And okay, this was one more topic that was left. So a graph, right? It's described by its nodes and edges. Nodes are these points, and edges are what are connecting the points, right? You can have directed graphs, which are pointed from a node to another node. So which generally means sort of a control or a directionality or regulation or control. And these nodes and these edges can either be all of the same size or they can be of different sizes depending on how important they are. Are they connected to more things? Do they have a higher effect and so on? So normally you would just, it is very similar to a correlation matrix. So let's say you want to find all versus all. You have 10 nodes and you want to see which 10 are connected to the other, right? So you would just write whether, so you compare node 1 and node 2. If they are connected, you write 1. If they're not connected, if they don't have any trend between them, you just write 0. That's what an adjacency matrix is. It just has 0 and 1 as its values. Some examples are protein-protein interactions, gene co-expression. That means which genes are expressed together, and which are gene regulatory network, which means which gene controls the other. Are they transcription factors, some splicing factors, or so on? All right. So Please let me know finally if there are any more questions. Otherwise, I think I have 5 or 6 questions that we'll quickly go through and see. Here, I would like each of you to answer. Okay, please answer. So this is going to help you in the assignment. All right. Okay. So which of the following will you use to set the range of values on the horizontal axis for a scatter plot? Right. We used it today multiple times. I don't want just one person to be answering. Okay. So B, which means xlim. So can any one of you tell me if I want to set xlim as between, let's say, 5 and 100, how would I write it? What would come within xlim? Quickly. We saw this today. Correct. Right? Very good. Okay. Now, which of this is an example of a directed graph? Gene regulatory, protein-protein binding, Facebook friends, network where atoms of a protein are represented as nodes and the bonds as edges. And whenever you give the answer to this for today, I would also like to have an explanation. Why do you think which of these is a directed graph? Please unmute yourself and just give a quick one-line explanation so that I know you've understood why it's. Okay, first give me the answer. Don't give the explanation. So a directed graph, that means one node points to the other. No idea? A. Can you also tell me why you think so? The answer is correct. So just let me know what points towards what. It tells which gene controls which gene. So it's typically a one-sided arrow. There are some cases where A gene controls B, B also controls A. So it can have a double-sided arrow. But yes, this is why it's going to be a directed graph. Protein-protein binding. So they're just binding. It's not one is coming to the other. Just both of them are binding. And Facebook friends, no, not typically a directed graph. And again, bonds, they would not be a directed graph. All right, good. So this is something we use today, right? Concentration and absorbance. So this is what we write. So which of the statements is wrong? One of the statement, at least one statement is wrong. Maybe 2 are wrong. I don't remember. So you can just tell me if the first statement is right or wrong. B, right? Because we're saying Y is absorbent. It says Y axis represents the concentration. So we know this is using the GG2 package. And we are not transforming anything. We are not calculating means, making a histogram, standard error, nothing. And it is a scatter plot because it says point. So yes, vertical axis represent concentration. You're all right. Okay. Okay. So here it says that the marks obtained by 11 students in exam. These are the 11 students, their respective marks. And if I use the R script to create a histogram to show the distribution, I'll write hist. And marks is, let's say it's a vector where I've saved. So marks is equal to C, all these values, and I write breaks is equal to 3. How many bars are going to be there? Give it a moment, think, and then I'll see if who was given the answer. 3. So breaks is equal to bins is equal to bars. So, right? So it's making 3 bins. So let's say it was from between 10 and 25. So it maybe made bins of 10 to 15, 15 to 20, 20 to 25. These were the 3 bins. Okay, good. And then we have which of the following R functions is used to add a curve which is fitting the data points. Smooth, point, abline, geomabline. So it is A, geom_smooth. Smooth. All right. So I think that's it. Done with today's class. So I will upload the recording of uh, today's session on YouTube, and the script I use and this PPT will also be uploaded to the Google Drive. The deadline for this week's assignment is day after tomorrow, 22nd, Wednesday. And we'll have the next live session next Monday, same time, where we'll be discussing week 5. All right. So yes, that's it. All right. I'll stop sharing. If any of you have any confusion, any doubt, please let me know. Okay. The best way to familiarize yourself with R is to practice. You have a lot of these data sets. You can try transforming these data, subset it, filter, and yeah, just make plots. You can see if more than one kind of plot from one data makes sense. Make these changes, explore all the arguments that are there within, and yeah, you will become experts at making these plots, right? And these come really very handy whenever you have any kind of data. The kinds of figures we discussed today, they are the most common ones, and they pretty much cover uh, all. So if you happen to take any research paper, you'll find one or more of these figures there, and you'll be able to actually identify if it was made using the default R functions or it was using ggplot, right? So I think that is all I have for today. Oh, the bar plot. Oh, good. Okay. All right. Let me share the screen again. Yeah. Okay. So here I wanted to make the same bar plot here, right? So we had this thing, right? This one. So we'll just write ggplot, and so the thing is, we'll have to make some changes to it, because if you remember when we used ggplot to make this, so one which was I, so it actually had, so what we are plotting is one of, so this is our x-axis and here it's the y-axis and so on. So yeah, so I think I'll also have to actually check how to make this. I'm pretty sure I will not be able to make this in the first go. So now to begin with, the F data frame does not have these as columns, right? So what I can do is yeah, I will actually try this after the session and I'll put the code in the script and then you can give it a try, because it will just take up, I think it'll take me 5-10 minutes. So I don't want to spend that time. Either way, both the functions are available with you, right? So, and actually you can also give it a try, right? I'll also try this. You also try this at your end and then let's see. Okay. Thank you so much. Thank you for joining. I'll see you all next week. And if there's any confusion, any problems, you can always put it on the discussion forum and you can discuss it in these live sessions. So thank you for being interactive. I hope from next time on, more of you are asking questions and giving answers. Okay. Thank you so much. See you next week. Bye-bye.